So what I want to talk about for the last 15 minutes or so is some really cool stuff that's new, that is actually far less sort of medieval and actually much more promising. And that's the frontiers of cancer treatment. And this is based on knowledge of oncogenes. And so what I want to talk about is a particular example. A disease called CML, chronic myelogenous leukemia. That is, in short, a blood cancer. That's what the leukemias are. There is an excess of myeloid cells. They're a kind of white blood cell. And that over time, it becomes acute instead of chronic. That leads to metastasis and eventually death. So the idea is that the leukemias kill you, so it's a tumor of blood-forming tissues. It's not a solid tumor, but you produce an excess of certain kinds of white blood cells. You think, why would that be bad? Well, first of all, they're immature. They're not the right kind. They're not useful as white blood cells to fight disease, is number one. Number two, it means your immune system isn't as strong as it ought to be, so you could be killed by an opportunistic infection. And number three, eventually they're going to start forming secondary tumors, metastatic tumors. They're going to metastasize to other parts of your body, and those will kill you. So it's bad news. Typically the way it works is it's chronic for a certain number of years, again, as mutations accumulate, and then at some point it goes into an acute phase, which if untreated is fatal. And for what it's worth, this killed my father in 1985. For several years it was chronic. It was treated for a while, but eventually it turned to acute, and we tried chemotherapy, but as in some cases it did not take. It's a killer of many people. And it turns out there's a couple of details that make it particularly nasty, but then there's also one of those details. It turns out to be something that they're now using as a cure that they didn't have back when my dad was sick. So let me tell you some of the details. There are many mutations involved. And it's tricky because there's a part of me that's like, ooh, this is cool biology. And then another part of me is like, it was my dad. And so at the time, I have to say I focused on the cool biology because I didn't want to think about the my dad part. And still, it's a mix. But with all of these things, they're real. We're not talking about some abstract trait in plants. You all will run into this as well. So I'm trying to treat it carefully, not just, ooh, cool, but remember there are people involved as well. So there are many mutations involved in this process, but there's one, the diagnostic one, it's called the Philadelphia chromosome. It's this very unusual thing, but it turns out to be common in this kind of leukemia. It's due to what's called a translocation. That is, DNA breaks and reconnects, which leads to rearranged chromosomes. And the way this works, it's actually a swap between number 9 and number 22. Now, why in particular these chromosomes? It may be that other swaps happen and they don't lead to trouble, but it happens that these particular chromosomes break in a particular place. It's not well understood, again, why, but this Philadelphia chromosome is found in almost all cases when CML becomes AML, acute myelogenous leukemia. And what happens is the DNA breaks. So cells have a mechanism. If a chromosome breaks, the cell's like, oh my God, that's not good. I got to fix it. And so it tries to find another broken end of a chromosome and glue them back together. Sometimes if one chromosome breaks, it joins it back. That's not a problem. If two chromosomes break, things can get tangled up, and sometimes the wrong pieces get reconnected. So when A breaks and B breaks, you actually get AB and BA stuck together. And so that's what happens in the case of the Philadelphia chromosome, is that in this case, it yields the following structure. If I would do it in two different colors. This is part of one chromosome. So this is part of chromosome 2 of chromosome 22. And there's a strong promoter. And there's a little bit of a gene called the BCR gene. And it's not quite known what that one does. But what happens is the BCR gene, there's a split, and it ends up joined up to most of the rest of another gene. This is called the Abel gene. And this here is part of chromosome 9. And this is a really weird kind of a mutation. This is not a point mutation. This is not an insertion. This is not a deletion. Literally, the two chromosomes break, and there's a swap made. And so the other half of this is going to be the front half of Abel stuck to the back half of BCR. It's a reciprocal thing. So it turns out that doesn't do anything interesting, so no one worries about that. So there was chromosome 22, the blue thing, and it broke right here. And chromosome 9, the green part, broke right there. And the cell was like, oh my God, I got ends. I'm going to stick them together. It sticks the wrong pieces together. It sticks 9 to 22 instead of 9 to 9 and 22 to 22. And when you do that, it turns out that and this fusion happens right in the middle of the coding region. The Abel gene is a growth control gene. And this, what you end up with now is this protein that's sort of like a bit of each. And they call it the BCR Abel protein. And you get lots of it because it's a strong promoter. And this thing stimulates cell growth. So the way this fits into CML and AML is there were some earlier mutations that caused some white blood cells to divide when they oughtn't. They're dividing more rapidly than they should, not super fast, but they're dividing fast enough to start messing things up. And at some point, what oftentimes this happens in mitosis, the division is happening so fast that the chromosomes get broken. And that chromosome break happens, and it's stuck together wrong. Sometimes it's stuck together in a way that doesn't cause any problems, but if it happens to get stuck together in a way that causes this structure, you make this BCR-ABL fusion protein. And that protein stimulates cell growth in a really big way. And the cells make a ton of it because there's that strong promoter. And as a result, these cells divide very rapidly. And that typically tips you over into the crisis mode where you have to get serious treatment then, otherwise you're in big trouble. Questions about that thing before we talk about how that yielded a really interesting way to treat the disease? About how that works. Yeah. Does this only affect bone tissue? Yes. So, or this is only happens in leukemias, apparently. All right. Other questions? Okay. So it turns out this thing is a target. This is a target. I'll actually do it in black. This is a target that's unique to cancer cells. The great thing about this protein is that it's not just ABL. If you inhibited ABL, that would have the same problem as chemotherapy. No one cares about BCR, but this is not ABL anymore. It's ABL with this big goober of something stuck onto it. And so that structure is unique to cancer cells. And it turns out that people designed to use the structure. So researchers use the structure of this BCR, the ABL protein, to design inhibitory drug called Gleevec. And it worked. And it was FDA approved in 2001. And so the key to this is if you inhibited ABL, it would lead to that would inhibit normal cells too. But Gleevec only inhibits the BCR ABL fusion protein. So that's why it works so well, because you can target that particular thing. And so there are all sorts of other drugs in the pipeline that are based on things like this. We now, because we know what's going on, we studied the biology, we can figure out a treatment that targets something unique to the cancer cells. And this works really well and has extremely limited side effects. Its limits are that blue-green junction doesn't always happen in exactly the same place. So basically, if it happens sort of too far to the right or too far to the left, you get a weird protein that Gleevec can't inhibit. So it only hits some, but it hits enough to make it worth doing and at least worth trying on most patients. And so that's an advance in the past 20 years or so that's made a huge difference in treatment for this particular cancer. The other trouble with this is it's one for one. It's one drug for one cancer, but that's how you have to do it one by one. Questions about this?